Assalamu alaikum everyone. So today we are discussing about a very important phenomenon in genetics, and we call that phenomenon as pleiotropy. And to begin with, we already know that there is normally one gene that controls the expression of one particular character. That means there is one gene and one character that are interlinked. But in certain conditions, the gene controls the expression of more than one character. And this condition where one gene controls the expression of more than one character or more than one phenotype, we call this condition as pleiotropy. To understand pleiotropy better, let's consider an example of a disease called as sickle cell anemia. By the term, we already know that anemia is a condition that is characterized by the lowering of the total blood content of a human being. What happens during this condition is there is the structure of hemoglobin that is altered, and there is a shape of RBCs that is altered. How that takes place? Before we dive into that, let's take a quick look on the normal structure of hemoglobin. Hemoglobin, as the name indicates, it consists of two parts, the heme part and the globin part. And this globin is basically a protein. We already know that proteins are made up of amino acids. Let's suppose there are multiple amino acids, and amino acids are connected together by the help of a bond called as peptide bond. Whenever there are multiple peptide bonds in a chain, we call this chain as polypeptide chain. We can infer from this that the globin, which is actually a protein, it is made up of polypeptide chains. Basically, there are 4 polypeptide chains that are present in this globin protein. Out of these 4 polypeptide chains, 2 are alpha chains and 2 are beta chains. In the condition of sickle cell anemia, what happens is one of the beta polypeptide chain undergoes point mutation. What does that mean? Let's say this is a normal polypeptide chain, beta polypeptide chain, and these circles, these represent the amino acids that are present in the beta polypeptide chain. These lines connecting the circles, they are the peptide bonds. Now, we have multiple peptide bonds, that means it is a polypeptide chain. In case of beta polypeptide chain, at position number 1, there is an amino acid called as valine. Position number 2, there is an amino acid called histidine. We have leucine, threonine, proline, glutamine or glutamic acid at position number 6, and the chain goes on and on. During point mutation, what happens is this glutamine at position number 6, it gets replaced by another amino acid called as valine. This is what we call point mutation. So there are basically 2 things that need to be remembered. First one is during the sickle cell anemia, there is the beta polypeptide chain that is affected by point mutation, and second thing is the amino acid present at position number 6 is replaced by another amino acid. Glutamine or glutamic acid is replaced by valine. As a result of these changes, what happens is, first of all, we know that the structure of the hemoglobin is altered, and we already know that hemoglobin is responsible for carrying oxygen to the body cells. Because of the change in the structure of hemoglobin, the oxygen carrying capacity of the hemoglobin gets reduced. There is a second effect of this change. The RBCs, which are normally biconcave in shape, they change their shape to sickle shaped, and hence the name of the disease. So we know that in case of sickle cell anemia, there is one gene that controls 2 expressions. First expression is the structure of hemoglobin, and second expression is the structure or the shape of RBCs. Because of the combined effect of the change in structure of hemoglobin and the change in the shape of RBCs, the overall oxygen supply to the body cells is hampered. And as a result, what may happen, since the person requires oxygen for the breakdown of food to release energy, any patient who is suffering from sickle cell anemia would experience fatigue or dizziness as a result of any gentle activity, because there is no sufficient oxygen available for the oxidation of food. Coming to the genomic representation of sickle cell anemia, HBS is the gene that represents the gene for the sickle cell anemic condition, and HBA is the gene that represents the normal healthy condition. Okay, and we can also remember that the genes that are associated with pleiotropy, they are called as pleiotropic genes. There are three conditions that may arise. Condition number one, a normal healthy patient can have HBA, HBA. These are the alleles, okay? And in condition number two, there can be one HBA, that is the healthy gene, and there can be HBS, that is the faulty gene. This allele would give rise to normal healthy RBCs and healthy hemoglobin, and this allele would give rise to faulty hemoglobin and faulty RBCs. As a result, the patient having this genotype would be having 50% healthy blood and 50% faulty blood. As a result, these patients would have sickle cell anemia, but they do survive with some deficiencies in their body, like there is deficiency of oxygen, as a result of which they might experience fatigue over gentle exercises. Third condition, when there is HBS, that means both the alleles are faulty or defective. This would lead to the formation of overall defective hemoglobin and overall defective RBCs. That means the entire blood would be defective. Such patients usually do not survive. But we also know that whenever there is any genetic disorder, Mother Nature compensates that in some way or the other. In case of sickle cell anemia, whenever any patient is suffering from sickle cell anemia, it's said that they are less likely to get affected by malaria. Why? Because the parasite that is responsible for causing malaria, that is Anopheles, that Plasmodium parasite coming from Anopheles is not able to survive in the sickle-shaped RBCs. That is why it is said any patient who is suffering from sickle cell anemia is very less likely to get affected by malaria. That was all about pleiotropy. Thank you so much for your time.